Well, let's continue with our discussion of laboratory techniques and the microbiology lab. We need to talk about our scope of size, our scale of size. If we look at most things that we can see with our naked eye, we can see meters pretty easily. Since most of us have been born and bred in the United States, we're used to the imperial or standard system. So things like meters, centimeters, micrometers, millimeters aren't necessarily very intuitive to us. But I've got to tell you, it's a better system. It's easier to work with if you do any kind of unit conversions. Because all you have to do is move a decimal point. So if we look at a centimeter is still very easily seen with the naked eye. We can see a microscopic organism that's as small as a millimeter. If you have good eyesight, you can definitely see something that is as small as a millimeter. Generally speaking, those are the little tiny lines on a metric ruler. There are 10 millimeters in 1 centimeter. Once we get to the scale of micrometers, that's where we need to use a microscope. The limitations for good eyesight for most human beings is going to be about half a millimeter or 500 micrometers. With a regular light microscope, we can go to typically as small as 5 to 10 micrometers. I take that back. We can go up to about 100 micrometers with easy eyesight for most laboratory grade microscopes. Anything smaller than 100 micrometers, you're typically going to want to use an electron microscope. Once you get to the scale of nanometers, you will absolutely need to use an electron microscope. Just so I can say that I said it, there are 100 centimeters in a meter, there are 1,000 millimeters in a meter, there are 1 million micrometers in a meter, and there are 1 trillion nanometers in a meter. So if we look at big or large eukaryotic cells, they're typically going to be 3 to 4 millimeters across. If we're looking at small, actually that's a typo. That should be micrometers across. If we're looking at small bacterial cells, we're going to have about 200 nanometers. That's about right. And then if we look at viron particles, we're typically going to have 20 nanometers to 800 nanometers. So if we look at this, the largest viruses are larger than the smallest bacteria. So things that we can comfortably see with our eye typically are in the range of 1 millimeter. If you have really good eyesight, you could see something that's 200, 300 micrometers across. Ooh, I should back up here. Most people don't have the the new symbol on their keyboard. So a lot of times in a healthcare setting, you'll see an MC as the abbreviation for the prefix micro. So if you see an MCM, that's a newer abbreviation for micrometer that has been popping up more and more over the last decade. Back to the figure. So things with a light microscope that you can see comfortably, our light microscopes typically go down to about 10 micrometers. Smaller than 10 micrometers, you're going to want to go with an electron microscope, and then to see individual molecules, individual atoms, so down to the size of individual atoms. This is what we have here. So this should actually say atoms, individual atoms, not amino acids. This smaller than 1 nanometer, is going to require very sophisticated imaging techniques. As we look at our microscope, the part you put your eye on is called the ocular lens or eyepiece. The lenses that you use to change your magnification are called the objective lenses. They are attached to the turret or the nose piece. The arm connects the head of the microscope to the base of the microscope and has the stage located in between. We have an aperture control or a diaphragm control that controls how much light gets to the specimen. And then we have the actual light source itself, which we are typically going to be able to have different filters go over, so we can have a green filter or a red filter to change the color of light. Most microscopes, electric microscopes, will have a small knob on the side or a dimming knob, plus a manual on-off switch. I remember the first classroom I taught in had old school microscopes with nothing but concave mirrors that you had to adjust. We could only use those microscopes if it was sunny out. And then I'd have my students sit in the sunshine, because that was the only light source we had that was bright enough, and the sunlight would bounce off the mirror and go back up into the specimen. To change our focus, we have the coarse adjustment and fine focus knobs, and then the stage adjustment knobs to move the specimen around on the microscope mechanical stage. When you are looking at your images on a microscope, we have the real and virtual images. That real image is formed by the objective lens. So we have those 3 objective lenses, sometimes 4 objective lenses. This scope has 3. And the virtual image is formed when that image is going to go through the ocular lens to us. So the real image is when the light goes through just the objective lens. The virtual image is the image that's gone through the objective and ocular lenses. Almost all ocular lenses are 10 times total magnification. To find the total power of your magnification, you multiply the magnification of the ocular by the magnification of the objective lens. So 4 times 10 gives us 40. 10 times 10 gives us 100. 40 times 10 gives us 400. And typically 400 times total magnification is going to be as high as you'll usually go with most light microscopes. If you're working with bacterial species, you need to have an oil on your slide, and you can use the 100x objective lens and end up with 1,000 times total magnification. The downside is you need to have really excellent optics for a 1,000 times magnification to work well. Most of the time, you're looking at least a $1,500 microscope for an image at 1,000 times magnification that you can actually make out. So here we have the light. That light source, or if we look at our typical microscope, we'll have the real image, which is the image that's gone through the objective lens. As that image goes through the ocular lens, it gets flip-flopped to form a virtual image, which is a mirror image of the real image, and that's what hits our eyeball and goes to our brain. Something that's very important in our microscopes is resolution, or resolving power. It's the ability to separate different structures from each other. Another way to think of resolution is how clear the image is. If it's very clear, then you can separate those objects from each other. But if it has low resolution, it's going to be really fuzzy, and it'll be hard to separate those 2 points from each other. Human eyes will typically have resolution of about 0.2 millimeters, or 200 microns. And that's if you have good eyesight. I was able to do that in college when I was looking at Arabidopsis thalianonectaries. I don't know if I could do that anymore. It's been a while since I've looked at something that small and had to resolve them from each other. Typically, most light microscopes that are using oil immersion lens can get down to as low as 0.2 microns, as opposed to the 0.2 millimeters. So most light microscopes have about 1,000 times the resolution or resolving power of the naked human eye. So let's do a what-if scenario. Let's look at the same thing with low and high resolution. Low resolution, it's very fuzzy, it's very blurry, and with high resolution, it's much easier to see distinct structures from each other. Unfortunately, getting those high resolution lenses, or the good optics, will cost most people a premium price. It's really easy to get 1,000 times magnification light microscope. And generally speaking, can be pretty cheap. I remember my first microscope would magnify up to 600 times, and it cost $20. It just had really, really low resolution. The optics were terrible, so you really couldn't see anything. Even though it was being magnified at 600 times total magnification, you didn't know what you were looking at. To get a microscope that can have high magnification and high resolution, That's something that's to be desirable, and that's something that's going to cost a lot of money. The reason we need to use oil immersion lenses is because when we are zooming in on really small areas or small fields of view with the 100x objective lens, we need to make sure as much light as possible goes in through the lens. When light goes through different mediums, from air to glass to water, generally speaking, that light is going to be refracted. It changes directions. This is the same principle that's behind a prism or an optical prism. When we use oil immersion lens oil, that oil sits in between the 100x objective lens and the slide that we're looking at, and it makes it so that the light does not bend as much. By using oil on an oil immersion lens, we make it so that more light goes into the microscope so that it's easier to see what we want to look at. When we're looking at our cells, if we use oil immersion oil, we'll get much higher resolution. So look on the right here, we can see two separate bacterial cells, or we can see our eukaryotic cell very clearly. But if we skip that oil immersion oil, it's going to be much more difficult to actually distinguish features because the resolution is going to be much lower. Something we need to keep be aware of is refractive index. As I mentioned 2 slides ago, refractive index is that bending or refracting of the light as it goes through different mediums. So some mediums will have light refract more than other mediums, and it depends on what 2 mediums you're comparing to each other. Ice compared to water, when light goes from water to ice, there's a very low index of refraction, but if you go from water to air, there's a very high index of refraction. The higher the difference in your refractive index, the more the light is going to bend. To change how much light can get into that microscope lens, we can use an iris diaphragm. And an iris diaphragm is different from just making the light bulb brighter or dimmer, because the iris diaphragm changes what selectively will choose what direction the light photons are traveling. If we have a very narrow diaphragm, we're going to have beams of light that are preferentially all traveling in the same direction, which is going to increase our contrast. If the iris is open wide up, that's going to allow beams of light that are traveling many different directions to enter the microscope lens, and that's going to reduce the contrast. There are several different kinds of light microscopes that we can use. A bright field microscope is the standard microscope that's used in most undergraduate laboratory practices. This is a microscope where we just have light shine through the sample into the lens. We also have dark field microscopes. Dark field microscopes are special because they highlight the special structures inside of the cells. These dark field microscopes are going to allow for the specimen to be illuminated from the side. So light will shine sideways through the specimens as opposed to directly through the specimens. We also have phase contrast microscopes. Phase contrast microscopes are going to allow us to see with greater clarity the structures, the organelles on the inside of our cells. Our phase contrast microscopes are going to cause our light to shine different wavelengths of light to have different preference as they're shining through the cell. By having those different wavelengths of light, we're going to have selective contrast as the light is shifting from phase to phase, from that liquid to the air phase. There's a lot of optics involved. This is not a physics class, so I'm not going to talk about the optical properties involved. Suffice to say that phase contrast and DIC, differential interference contrast microscopes, both involve using different wavelengths of light being shined at different angles through the cells to highlight the internal structures of the cells. Phase contrast and DIC microscopes are pricey. They are very difficult to get a hold of. Fluorescent microscopes are fairly straightforward. A fluorescent microscope is going to shine fluorescent light, ultraviolet radiation, through the lens, and this is going to cause proteins or other chemicals within the cells to start fluorescing. And we can change the wavelength of ultraviolet light to change what kind of fluorescing we have occurring in the cell. A confocal microscope is going to be able to take an image at different planes of field along the z-axis. A confocal microscope can be combined with a fluorescent microscope to allow you to cause the cell to light up or different parts to fluoresce different colors, and then you can take different pictures at different slices throughout the cell. Sometimes it's referred to as a z-stack as we are working with the cells. A confocal compared with a fluorescent microscope gives you lots of those really cool GIF images, so you can see different layers of the cells within a slice of tissue. Then we get to the transmission and scanning electron microscopes. And once we get to these guys, we now jump down to the resolution range of nanometers. Up until this point, with light microscopes, we've been all the way up in microns or micrometers. But now we can add an order or several orders of magnitude, 3 orders of magnitude, and jump down to the nanometer resolution range. A transmission microscope is going to blast electrons through the sample, and we look at how the electrons change as they move through the sample. And then a scanning electron microscope, instead of blasting electrons through the sample, it's going to bounce electrons off the top of the sample. The reason that we're able to have much higher resolution with electron microscopes compared to light microscopes is because electrons oscillate at a much smaller wavelength than photons of visible light. Photons of visible light oscillate at relatively large wavelengths compared to electrons, which oscillate at incredibly small wavelengths. So, enough about microscopes. Let's talk about how to get the specimen rated beyond the microscope. In almost every situation, you need to take that specimen and put it on a microscope slide or a glass slide. And that slide is going to sit between the condenser that shines the light and the objective lens, which receives the light. Some things are going to be very important, though, and will influence how you prepare your sample. Is this specimen alive or dead? What is your goal? Do you want to see the overall big structure? Do you want to identify the structure, the cell type? Or do you want to see how it moves around? So this is going to impact what kind of glass slide you choose. Also, another thing that will impact is what kind of microscopes you have available. Do you have dark field? Do you have bright field? Do you have electron microscopes? Do you have fluorescent microscopes? So the equipment you have available, what you need to see, and what kind of specimen you have to work with are all factors that influence how you prepare your microscope slide. If you're working with a fresh or living preparation, you're typically going to use a wet mount or a hanging drop so you can observe the organism in as close to its natural state as possible. These cells, or these microscope slides, if we have a wet mount, are typically going to have the drop on the underside of the microscope slide. Sometimes we'll have special microscope slides with indentations in the middle that can have a pool of water through them. I prefer those because it's easier to work with that. You don't have to worry about water getting all over the place. When we make a wet mount microscope slide, you'll take a drop or two of the liquid medium that contains your microorganism, put it on the top of the microscope slide, and then put a very super thin piece of glass over the top of that called a cover slip. So you make a glass sandwich with a drop of water in the middle. A hanging drop, on the other hand, is going to have that drop of water typically on the underside of the microscope slide, or if you have nice microscope slides, hence the name hanging drop, you used to put it on the underside of the microscope slide. But now we have those microscope slides with the indentations in them, or the concave microscope slides, and then you put a cover slip over the top of that. So we can have a hanging drop specimen. Sometimes that hanging drop specimen is going to be hanging off the cover slip that you put over the concave indentation. Usually, though, in most situations, you'll see the specimen just drop directly in the pool and then have the cover slip put over the top. And this entire pool or depression is filled with liquid. When working with light microscopes, particularly bright field light microscopes, it's tough to see the inside of the cell. It's a big wash, a big mess. So we need to use special chemicals that make it so that we can discern the structures inside of the cell. And those special chemicals are typically going to be a stain. The process of staining is any procedure that applies colors or chemicals to the specimen that you're looking at on the microscope slide. We can have basic dyes. Basic dyes, generally speaking, are going to have a positive charge associated with them. And depending on what kind of microorganism you're working with, you'll want to use either a basic or an acidic dye. Acidic dyes have negative charges. If we look at bacteria, particularly gram-positive bacteria, they have lots of negatively charged groups, macromolecules on their surface. So if we use a basic dye with a bacterium, generally speaking, a basic dye sticks really well to the bacterium. But let's say you want to see the overall shape of the bacteria. You don't want to see structures in the bacteria, but you want to color the background. If you want to color the background, you should use an acidic dye, because the positive charge of the acidic dye is going to be repelled from the positively charged bacteria. Some stains are going to stick to our specimens. Those are called positive stains. With bacteria, most basic stains are going to be positive stains. If the stain doesn't stick to the bacteria, the microorganism, but sticks to the microscope slide, the background, we call that a negative stain. For most bacteria, acidic stains are negatively stained, or are negative stains. When performing a negative stain, it's very, very important to not heat fix the cells to your slides. The flaming process from heat fixing the cells to the microscope slide with the Bunsen burner is almost always going to damage the structures that you're trying to look at. Two very common dyes that are used in this negative staining process are Negrosin stain, or Negrosin dye, or India ink dye. We could also have a simple versus differential staining. As its name implies, a differential stain is used to tell the difference between microorganisms. We'll have a primary stain, which is given first, and a counter stain, which is used to color the negative cells that the primary stain does not stick very well to. We can use differential stains for different cell types, the most popular of which is gram-positive versus gram-negative, the gram stain. These differential stains are going to typically be much more complicated than our simple stains, and are going to involve multiple staining steps or chemical washes. A simple stain is, generally speaking, a two-step process. You put the stain on the microscope slide, and then you rinse the extra stain. That's step one. And then step two is rinse the extra stain off the microscope slide. You use simple stains if you just want to see simple things about the cells, like how many cells are there? What's the size of the cell? What's the shape of the cell? What is the arrangement of the cell? Whereas differential stains can tell you all that, and also tell you information based on the chemical properties of that cell. So here's some classic simple stains: crystal violet and methylene blue. Here's some bacilli, E. coli cells that have been stained with crystal violet. Here is some Corynebacterium, so we can see some nice dark blue cells that have been stained. That gram stain is an oldie and a goodie. It was developed in 1884 by Hans Christian Gram, and it has 4 primary steps. Step 1 is to expose the cells to crystal violet, our primary stain. Step 2 is to add iodine. Iodine acts as a mordant, and a mordant is any kind of chemical that makes the dye stick. For those of you that are into dyeing your own wools or making your own fabrics or knitting a scarf or a sweater and then dyeing the scarf or sweater, you typically will use a mordant to make the dye that you color your fabric and make that dye stick to the fabric. And we use mordants in microbiology to cause the dyes to stick to the bacterium. So iodine causes crystal violet to stick to gram-positive bacteria, but not gram-negative bacteria. Crystal violet will initially color all the bacteria dark purple. Then we rinse it off, add the iodine, and the iodine causes the crystal violet to stick really tightly to the gram-positive bacteria, but not the gram-negative. Then we use an acetic alcohol solution, or an acid alcohol solution, and we rinse it off. And the crystal violet rinses off of the gram-negative bacteria, but it's still stuck to the gram-positive bacteria. And at this point, if you were to look at a microscope, if you did it correctly, you should only see gram-positive bacteria, because the gram-negative bacteria don't have any color to them at this point. So to see the gram-negative bacteria, we add safranin, which is a kind of a light pink stain. It's the counter stain. Gram-negative bacteria appear pink, and the gram-positive bacteria will have that dark purple, the crystal violet stain sticking to them. So let's skip ahead here. Here is a good example of a gram stain. You can see the nice pink gram-negatives and the dark purple gram-positives. Oof, that is a beautiful gram stain. They almost never appear that beautiful. Our differential results in the gram stain are indicative of the differences in the outside of the bacterial cells. A gram-negative cell has a phospholipid layer on its surface that makes it very difficult for the crystal violet to stick to, whereas a gram-positive cell has a thick layer of peptidoglycan on its surface, which is very easy for the crystal violet to stick to. The gram stain is one of the gold standards in bacterial identification. When you're talking about a human being, you talk about vital stats like age, weight, gender, hair color, eye color, the vital stats you see on your driver's license card. If we were to make a driver's license card for E. coli or Clostridium difficile, if we were to make a driver's license card for a species of bacteria, one of the things you would include on that is it gram-positive or gram-negative. Gram staining the gram staining is one of the vital stats for bacteria. Another one is our acid-fast stain. Our acid-fast stain will differentiate acid-fast from non-acid-fast bacteria. If they're acid-fast, they are going to be stained pink, whereas if they're non-acid-fast, they'll be stained blue. The acid-fast stain is the gold method, or gold standard, for finding Mycobacterium tuberculosis, or the bacteria that causes the disease tuberculosis. The genus Mycobacterium is well known for having a very thick, waxy glycocalyx. And this thick, waxy glycocalyx has different chemical properties compared to most other bacterial species. Mycobacterium tuberculosis is one of the few bacteriums that is gram-indeterminate. It's not gram-negative. It's not gram-positive. It's gram-indeterminate. Safranin and crystal violet do not stick to Mycobacterium tuberculosis because of its thick, waxy coating on the outside. But something that does stick to it is carbofusion. Carbofusion will stick very tightly to it to give it a pink color, and then the other bacteria that are present will have a dark blue color when they're stained with methylene blue. We can also use the acid-fast stain for other species of bacteria, fungi, and protozoa, but it's almost always used for Mycobacterium tuberculosis. So here we have some acid-fast staining. So we can see the pink cells are kind of blurred and blobby looking, and that's because they have that big waxy coating that the carbofusion is sticking to. And then you can see the blue cells in the background. Those blue cells that are in the background are non-acid fast. They typically do not have that thick, waxy coating associated with them. We also have the endospore stain. Our endospore stain is very similar to the acid-fast stain, except that we need to get the dye all the way into the center of the cell. This is typically going to be done with a dye known as malachite green, and this endospore stain is one of the more difficult staining procedures that you'll ever perform in a microbiology lab. One of the reasons for this is because it's more of an art form. And I should say that all staining procedures are, in some sense or some degree, in art form, but particularly the endospore stain, because it involves steaming your microorganisms and air drying and air fixing, and there's lots of extra steps involved with this procedure. You'll find out when you do your online lab activities. This endospore stain will differentiate between a cell that's a really old bacterial cell that has a spore on the inside, or a cell that's rapidly reproducing, a vegetative cell. Most of our endospore staining is going to be done to identify endospore forming bacteria and the Bacillus and Clostridium genii of bacteria. And it's kind of hard to see on this screen. I can't really zoom in right now, but you can see a pink bacteria, and in the very center of some of the pink bacteria, like this one right here, it bulges out in the center and there's a green dot because there's an endospore. But oh, where's my mouse? There's my mouse. But over here, there is no bulging green dot in the middle, so these are going to be vegetative cells. And then here's a cell that's dormant with an endospore in the center. We can also have some other special staining techniques. These are used to emphasize parts of the cells that are typically very difficult to find with conventional staining methods, such as the capsule staining technique. Some microbes will have a microbial capsule. This is a big, thick layer of wax in the glycocalyx that surrounds some bacteria and fungi, Mycobacterium tuberculosis. This is going to be negatively stained with India ink. Or we can use a flagellar staining technique. Flagellar staining is used to reveal the presence of a flagella. A flagellum is a long extension from the cell wall and cell membrane of the bacterium. Flagella are used for locomotion, and this flagellar staining is going to coat flagella based off of their electric charges. Are they positive or are they negative? We will use a stain that sticks to that specific structure. So here we can see India ink staining. So this is going to be a negative stain. We stained the background of the microscope slide, and then there's the bacterial cell in the center, and then around the bacterial cell is that thick, waxy glycocalyx that's pushing the India ink away. So here we have Cryptococcus neoformans. I absolutely would not want to be infected with Cryptococcus neoformans. Those thick capsules that they have would make it very difficult for my body to fight off an infection of this bacteria. Here's Proteus vulgarius. Proteus vulgarius is notoriously known as the bacterial species with lots of flagella. And if we look at Proteus vulgarius, most microbiology labs keep a couple samples of these on hand at all times, just as a positive control for the flagellar stain. We can see a bacterium that has lots and lots of itty-bitty tiny flagella coming off of it. And if we look at these little bacteria over here, they also have lots and lots of flagella coming off of them. So, by the end of this chapter, you should be able to identify the staining techniques. Each of these staining techniques is simple, differential, or special. We have a capsule stain, acid-fast stain, crystal violet, gram stain, and flagellar stain. Capsule stains are differential. They separate cells that do and make capsules from cells that do not make capsules. The acid-fast stain is also differential. Our acid-fast stain is generally speaking used to separate Mycobacterium tuberculosis from other bacterial species. Crystal violet is simple staining. It just stains the entire bacterium regardless of the bacterium species. Gram staining is differential. Gram staining is going to determine if it's gram positive or gram negative, or, heaven forbid, gram indeterminate. I hate it when that happens, but it does every once in a while. And then finally we have flagellar staining, which is one of our special stains. That's all we have for this chapter. If you have any questions, please feel free to shoot me an email. Happy studies!